ערב טוב לכולם, ברוכים הבאים. נמצאת איתנו היום דוקטור גרוסמן, שהיא סגנית נשיא בחברת טבע, ראש המחלקה לרפואה מותאמת אישית וביג דאטה, בתוך חטיבת הפיתוח והמחקר העולמית של טבע. איריס הקדישה את קריירת המחקר שלה הן בתעשייה והן באקדמיה לקידום של מחקר של רפואה אישית. לפני שהצטרפה לטבע היא ניהלה את ישראג'ין, חברה לייעוץ וניהול לחברות פארמה למגזר של ביוטכנולוגיה, והיא בנתה תוכנית עבור חברות פארמה גנטיות, היא עברה לייעוץ אחרי שהייתה אחראית להפעלת תוכניות פארמקולוגיה בקנה מידה גדול ב-GSK, תוך שימת דגש על מחלות זיהומיות נוירולוגיות. באקדמיה, איריס, דוקטור גרוסמן, השלימה את לימודי הרפואה בטכניון בשיתוף עם המחלקה לגנטיקה מולקולרית במכון וייצמן, והשלימה פוסט-דוק באוניברסיטת דיוק במרכז של פרופסור דוד גולדשטיין. לאחרונה הצטרפה איריס לוועדה המייעצת של סייאנס אברוד, ואנחנו מאוד גאים בזה ומאוד מודים לה על כך. איריס, הבמה שלך.
תודה רבה סיגל, זה באמת כבוד גדול.
שומעים.
זה בהחלט כבוד גדול להיות פה. אני רואה בעצמי עדיין בוגרת בחלק מביו אברוד, סייאנס אברוד בעצמי, בפוסט-דוק ואחריו בנורת' קרוליינה, אז בהחלט looking forward to participate and contribute also on the board. So without further ado, I'd like to share with you what personalized medicine means in the context of industry and what we do, and hopefully maybe even spark interest to come to us or to others like us. So I want to start from the end. I want to start from the concepts of personalized medicine, and before we define the terminology, it really is all about a holistic healthcare management. It's about looking at individuals throughout their life cycle and throughout their needs from the time that they're being that they could be identified at risk via whatever methods, through the time that they can be diagnosed early for their symptoms, the contribution and constellation of the different morbidities that they may experience in concert, and eventually increase the precision of our tools to provide different individuals with the right medication at the right time, at the right dose for each and every one, or for smaller and smaller patient populations. And then other aspects of personalized medicine or holistic healthcare management include the management of combination and repurposed therapies, but also non-therapeutic solutions like diagnostics, like monitoring tools, predictive algorithms, which we will talk about quite a lot later today. And so let's start from the beginning now. What is personalized medicine? So if you ask the FDA in one of the recent publications, the terminology is the tailoring of medical treatment to the individual's characteristics, needs, and preferences of the patients. So the characteristics, needs, and preferences can be measured by many different modalities, and specifically in Teva, these are some examples of what we are already doing in the last several years. So first and foremost, we collect DNA from all patients in all clinical trials. This is a mandatory aspect of how we serve the program. Of course, there are other labs and other measurements, clinical measurements that are taken, And now we've started to use different sensors and different other modalities as well. So you can see here on the bottom in the CNS, in the central nervous system, we of course use imaging, sleep monitors, movement disorder monitors, and we'll talk about some of those in a minute. In the respiratory space, so these are the two main therapeutic areas that Teva R&D focuses on, the CNS and respiratory areas. Of course, in respiratory, we have different relevant markers, whether it's imaging and other respiratory measurements. And then kind of converging again at the end, another quite ubiquitous marker, just like we all have DNA. Pretty much all of us have a cell phone these days, and that can also be used as a very useful marker in different constellations. And so overall, in fact, we live in a fantastic era. There are so many different types of markers that we could harness and look at. Many of them are validated, and the functionality and relevance is becoming more and more clear and direct. But really, what one of the main messages that I want to convey is, at the end of the day, and at the beginning of the day actually, it's all about the scientific question. It's all about what is it that we're trying to capture for each and every program. And then we tailor the right modality or several modalities. But you will see in the programs that I will share with you today, each has its own tailored approach, because we really want to make sure that we're not just shooting in all directions and then being left with all the false positives. And so just to bring it down to a very concrete level, before we go into what specifically my group is doing, I just want to make a very clear message that personalized medicine is certainly not just a vision or a nice futuristic concept. It is today and actually yesterday. And I'm not going to talk about the specific examples in this or the next slide. I'm just trying to convey that probably each and every one of us has encountered personalized medicine tools in our healthcare or the healthcare management of our loved ones. So these are just a few of them. Again, I prefer to spend time today on concrete examples from our pipeline. But if there are any questions later, I'd be happy to address them. I also want to make it very clear that for us as the pharmaceutical industry, there are many incentives to use personalized medicine. I often get questions, why would a pharmaceutical want to limit its market? And in fact, it's not about limiting the market, it's about providing true value, outcome-based value to patients. So increasing the efficacy and reducing the safety profile of our medicines, not to mention the ability to increase the probability of success, which we'll talk about in a minute. And as you will see in the next couple of slides, the regulators also feel very strongly that this is what we should be doing. Here in this report from more than 3 years ago already, FDA already then talked about 15 years of them paving the way of personalized medicine, and with them, other regulatory agencies have been very proactive. And you can see that back then already about a third of all submissions, all the drug programs that were submitted to FDA already then included a component that is key to the efficacy, safety, or pharmacokinetics of the drug based on biomarkers. And same goes for the EMA. I'm not going to talk about that right now. What it is important to mention actually in this context is that just about 13% of all marketed drugs in the US and a little bit more in the EU have biomarker information on the drug label. And so by indication, the regulators expect the physicians to use this information in the way that they prescribe medicines. And that trend is getting stronger and stronger. These are the many types of guidances that the various regulators worldwide have issued, which again show how much involved and leading they are in this field, and again in their expectations out of us, from us as the pharmaceutical industry. And this is how the FDA views the value of personalized medicine across the entire pipeline. And this actually very well represents what we are doing across the pipeline of our products at Teva. Right from the very early stages, discovery, non-clinical, here on the left, we use different modalities to understand the mode of action of the drug, metabolism, consider safety issues, and indications, and then move on along the development plan and up to post-marketing in the phase 4 programs. What you can see here on the very top, as we go along methodologically through these steps, we optimize the efficacy and minimize the risk of our products. And I think this is the last kind of landscape slide that I have today, again, to ground us on the true value of personalized medicine. It's not just about going through the development process. It's not just about getting approval by regulators. But today, more than ever, it's all about getting the right value for investment, the return on investment. And so for a drug to be truly successful, it needs to be reimbursed. It needs to be high on the treatment algorithm that physicians use. And so here are 3 examples. I'm actually not going to go through much details, but I've been involved in several of these, and the point was to show that with the increased benefit to risk ratio that we can provide once we account for, in this case, genetic markers, we can position the drug in a way that brings value to more patients in a more substantial way. And these are all these examples took different approaches around this exact question. And I took those because one is in HIV, the other is in cancer, and the third relates to anticoagulation. So very different therapeutic areas, and all see great value from these approaches. And so all in all, more and more we see that the use of personalized medicine approaches, the integrated analytics, and the multi-marker approach is enabling us to go between different stages around development and marketing, and LCM is life cycle management, where you leverage the product to other indications or other uses. And you can see that by being able to have this reciprocity around this circle, we are able to provide value to all the stakeholders, whether it's patients, physicians, payers, and so on. Okay, so all this this lovely introduction, what does it mean for us, specifically in Teva, and potentially to you? This is the mission statement of my team, Personalized and Predictive Medicine, Analytics, and Big Data, this long acronym here. We see ourselves as the stewardess of creatively leading innovation in Teva R&D through state-of-the-art personalized medicine strategies that characterize, position, and differentiate our medicines, as well as delivering sustainable, holistic solutions derived from biological and digital marker-based analytics. Okay, let's see what that means. We, in Teva, as I mentioned earlier, work mostly in the CNS space and in respiratory diseases. The main areas are migraine and headache, Huntington disease, multiple sclerosis, asthma, neuropsychiatric disorders. And throughout the pipeline, we operate. These are the types of questions that we focus on around these assets. So if we start from the top left and go around, we ask questions about how can we identify responders to our medicines. We'll talk about a specific example in a second. Then we look at ways to identify safety events, predict them, and exclude patients who are prone to develop safety events. We use these modalities to defend and differentiate our products. Again, we'll talk about an example. Of course, one of the major hallmarks of personalized medicine is being able to translate biomarkers from the discovery stage into the clinical stage. As we all know, not everything that we can detect in the animal or preclinical model has direct relevance to the human, but many things do if you apply the right approaches and considerations, as well as analytical approaches. Repurposing, as I mentioned, we'll look at later on, and then lastly, prognosis of disease progression and attacks. This is a bit of about us. My team is a small team. I started this unit at Teva about 3.5 years ago. At the time, we were 2 people. Now we're just about 11 people. And you can see on the top 4 boxes are characteristics of this team. Everyone is a PhD with either postdoc or corporate or small biotech experience, and we put a lot of emphasis on publishing. We're it's very important to us to keep up to speed with the cutting edge research and collaborate. And so we have one of the relevant measurements is 11 peer-reviewed publications since we were established mid-2013, and that doesn't include all the manuscript we're working on. And one of the most important elements of how we operate, particularly in the personalized medicine space, but also in broader Teva and particularly R&D, is collaboration. And what you can see here are all the different partnerships that characterized us in 2016. Some of them on the very left are academic based. We have a network of excellence of research in neurosciences, the NNE, which, as you can see in this little figure, creates collaborations between us and virtually all academia in Israel, as well as between them, and facilitating a very unique ecosystem where the collaboration is very much around reciprocity, not just, but of course, including grants. We are part of the IMI, which is a big consortium in Europe that funds research that is pre-competitive, together with the European Commission, enables us to really reach out very broadly to many academic and small companies to help us in these research initiatives. And then we work with a lot of other companies. We'll talk a lot today about Immuneering, which is a company that we work with very closely. In fact, since the first day I joined Teva, we've had this collaboration working specifically on analysis of genomic data and a very broad and deep framework, a lot of innovation, both in terms of methods as well as in application. And Immuneering sits in Boston. IBM Watson and IBM Research are two entities, very different entities that we work with as well quite closely, and I'm not going to go through all of these, but we've incorporated a lot into the way we work. So a bit of a step back about us as Teva R&D, so that you can appreciate the examples that I'm going to focus on in the next several slides. So in general, where we see R&D in the central nervous system area, it's quite complex. It might be one of the most challenging therapeutic areas for a company, for a pharmaceutical company to go after. But we are strong believers that this is where the unmet need is, and this is where expertise and deep scientific understanding really matters. We look at commonalities of neurodegenerative disease pathways. We'll see an example of that. Early predictors, novel biomarkers, early regulatory involvement, and leveraging external innovation. Just to make sure we're all appreciate why CNS is part of our DNA. Teva is already a leading CNS competitor in the US. We are global market leaders in multiple sclerosis with Copaxone, in wake or sleep disorders with Nuvigil, Parkinson's disease with Azilect. We all know both Azilect and Copaxone originated in Israel, the Technion Weizmann Institute. And we have a very broad and deep set of capabilities worldwide. We are also the largest generic company in the world, so we have a very strong distribution network. We are highly committed to the CNS space, and we focus on each and every one of the areas that I'm listing here. We're in fact the #1 developer in Huntington's disease, which is a rare disease, but we have 3 different development programs, including 2 at very late stages, which we're very excited about. Okay, so there are going to be 3 themes that I'm going to talk about as I go through examples from my pipeline. The first talks about evidence-based approach increases the probability of success of our products. And here what we see are 2 pieces of information from published data. The bottom says, basically shows the rate of success of a clinical program. So in phase 2, probability to succeed is 32%, in phase 3 61%. So it's a very high risk area to be in. But when we look at the reasons for failure, we see that for the most part, the failures are attributed to lack of efficacy. And what this tells us is that probably, as a whole, the pharmaceutical industry has not been critical enough and ensured that the drugs that we develop are actually conferring efficacy. Maybe they're not targeting the right targets, or targeting them in the way that we expect them to. And this and the value of the increasing the probability of success or losing the probability of success scores can be quite substantial. For instance, a phase 2 study, if we increased the probability of success by just 15%, it could be, on average, a general calculation, it could mean $500 million of investment. So this means a lot. And what by now 3 different publications of very large meta-analysis done by different companies show that when you have genetic validation of your target, you are twice as likely to succeed in your development plan all the way through to launch. So this has a lot of relevance. And of course, we at Teva are harnessing these approaches in many or almost all of our drugs have very well characterized targets. And I'm going to talk about just one example, one that I like specifically, because I think it is a great depiction of all these concepts that I just mentioned. So it pertains to the management of pain, and it really starts from epidemiological studies that were done, in a different era when epidemiological studies took a long time. But what we can see here is a continuum of pain phenotypes in basically the distribution of pain phenotypes in the population. This is the Gaussian here. If you look all the way to the left, you see individuals like the human pincushion, which depicts individuals that experience no pain. They can feel heat and touch and pressure, but not pain. And they're very rare, but they exist around the world. And if you look all the way to the other extreme, there are individuals that experience spontaneous exacerbations of pain, and it's excruciating pain. The triggers can be very trivial. Many of these patients say that they cannot take a hot shower, that wouldn't be something they can bear. And then, of course, the rest of us sit somewhere in the middle, probably in the middle of that Gaussian. We are a little bit more, a little bit less sensitive to pain. And if you look at the genetics of these individuals, you will see that mutations that cause lack of function, gain of function, are mutations that characterize individuals with spontaneous pain, and individuals that have loss of function mutations actually do not feel pain. And all of these mutations have been found in a single gene. It's an ion channel, and of course the lack or gain of function translates to the firing of this channel. And what's beautiful about this is that very rare phenotypes led us to identify a whole new target for pain management that could, if phase 3 trials succeed, completely revolutionize the way we manage pain in a non-opioid mechanism that is sorely needed in pain. And we also know, thanks to all these epidemiological studies, that although the initial findings were in very, very rare cases around the world, they matter to the general population, because we do see common variants, including the R1150W, which is associated with different sensitivity to pain in the common population. The last most important aspect of these kinds of approaches, discovery approaches, is that the individuals with those rare mutations that completely change the way this channel works the individuals are otherwise normal. They live their lives as normal human beings with very little other abnormalities, which means that by targeting this gene, by inhibiting it, we are not likely to induce safety or mechanistic safety issues, which is, of course, a critical point for development of drugs. And in fact, when we looked at the phase 2 study results for our own NaV1.7 inhibitor, we could identify preliminary results that show that individuals that carry this common variant, the one that is common in the population and affects sensitivity to pain, the carriers of the rare variant are actually twice as likely to respond to therapy, which again is a huge increase and very relevant to the way we are already developing this drug. The second theme, and here I'm going to go through very quick examples, relates to characterization and differentiation of our anchor medicines in different areas. The first one that I'm going to talk about, and I'm going to use our publications as a lead to the story, and I invite you to look at those. This is a medicine that we are developing for Huntington disease, one of those I mentioned earlier. And what you can see here is a recent publication, again with Immuneering that I mentioned earlier, and a whole host of internal collaborators from discovery, pharmacology. We work very closely together. And what you can see here is a very elaborate and very exciting network of new pathways that we identified for this drug. And on the right are the gene expression findings, and on the left is a depiction of validation for the BDNF secretion levels. And what you can see here, and I hope it's not too busy, this slide, but on the top, when we bought predopidine, we bought it from a different company, assuming or with the knowledge that it is a dopamine stabilizer. You can see that in the very top. But we had a sense that the clinical phenotype that we saw from the earlier phase 2 studies that the other company did likely were not explained just by dopamine stabilization. We decided to go and dig deep to understand the mechanism of action. And here in the middle, you can see the variety of studies we did with external and internal collaborators, looking at a whole host of different modalities. And again, gene expression is a major component here. And what you can see on the bottom is that we identified at least 4 major new pathways, mechanisms that show the possibility, a great potential for this drug to affect not only Huntington, but other neurodegenerative diseases, because it has neuroprotection effects. It could improve cognition and, of course, motor function, as we see in all the preclinical models. And now we have phase 2 study results that are highly supportive, and we're now going into phase 3 in Huntington, but already looking at least 2 additional indications as we speak. All thanks to this very deep understanding of the science. Again, this is another publication. I think the last example in this section relates to some of the work we do for drugs that are in the post-marketing stage. And this is Copaxone. I'm sure many of you heard about it. It's a multiple sclerosis drug that we developed and licensed out of the Weizmann Institute. And what we have, it's a very unique compound. It's actually not a small molecule. It's not a biologic. It's a very complex synthetic mixture of polypeptides, actually up to 10 to the power 29 different variants in the mixture. And that creates a very, and it is immunogenic. It works through the immune system, it affects the T cells. And what we can see is that when we apply different novel methodologies that we develop and publish extensively, as well as genomics, we see differences between Copaxone and the follow-on medications that now enter the market in different geographies. And our data shows impact on functional and immunological pathways. There are reports from patients in Mexico that talk about increased adverse events. And this poses a very important tool to understand how and what to do in terms of development of follow-on products. think last example is another compound, laquinimod, which for many years we did not know what was the actual target of. We knew it affects multiple sclerosis. It's an effective. It has shown efficacy throughout development. It is now in phase 3 clinical trials, but only through very intense recent efforts could we identify the target, and a big part of the support to that has come from again gene expression and other efforts as published here in PNAS last year. Okay, the last thing we're going to talk about is big data analytics as a boost to the pipeline, and I'm going to share here 2 examples of how we apply this capability, but we use it in a much more ubiquitous way. The first relates to repurposing, a major effort that I truly believe Teva has a unique advantage in because of our big pipeline, the big cabinet of medicines that include generics, but also very significant specialty development efforts, which are very rarely combined together the way they are in Teva. So what repurposing allows one is to reduce the development timeline, because marketed drugs already have a well-known safety profile, the preclinical package is known, there are many elements that one can rely on that have already been established, and basically jump into the later stages and pursue a shorter development plan. Now, it's important to note that repurposing is certainly not a novel concept. In fact, as you can see here in this cheat sheet, about 30% of new market entrants are in fact repurposed drugs, including kind of life cycle management of different medicines. And that translates into value, because about a quarter of annual revenue is accounted for by these drugs. As I mentioned, the real draw to go after repurposing is that we know a lot about the drugs already, and the most important aspect is safety. As we talked earlier, many drugs fail due to safety issues that are much less likely with a repurposed drug. Then we talked about the reduced timelines potentially. And so if it has been done for so long, why do we think we're special or new, or what's new about today and now? Well, if we look at history, and you can see there's some repurposed drugs as early as the 1950s, those instances, as we look along this trajectory, have been mostly around single effort, serendipity, maybe focused effort by a particular developer. But what we're doing today with the advent of big data, use of analytics, use of different mechanistic understanding efforts, is systematically approach repurposing in a very wide net approach, in a very systematic depth and very innovative way. And we have several different approaches, but the real, the main effort and the focus has been through genomics-based approaches. So again, we're great believers in mechanistic understanding of drug and the concept that most drugs are actually were developed for a particular indication, a particular pathway, but actually affect many others and hold great potential for other indications. And so with Immuneering, we've been developing an engine that looks at these opportunities, which has been increasing its its yield and substantiality. And then another collaboration that we started late last year was, I think in the news around October, is a unique partnership with actually IBM Research and IBM Watson here in Israel, where we're developing an engine, a repurposing engine around real-world data. And then in the area of analytics and digital markers, we are looking at a much broader net from different gadgets and all the way through social media in a very patient-focused manner. I'm going to give you two examples before I'm approaching the end of the talk. This is an example. We again published a poster earlier this late last year about a collaboration with Intel in Huntington disease, and this is basically we have a pebble watch, and we're using the accelerometer to measure the movement disorder of patients and develop algorithms that provide state detection, which is a much more accurate measure tool for understanding disease, for increasing the accuracy and reliability of the way we do clinical trials, and hopefully once validated, it would also be useful for patients and physicians, and potentially even a marker for response in the future. So this is an exciting activity. The other is around e-connectivity capabilities. We bought Gecko Cup, I guess already 2 years ago, which allows us to have connectivity capabilities on our inhalers in asthma. And we are developing, again, different algorithms to provide more meaningful and actionable content to these inhalers. So I reach my summary. Molecular and digital understanding of disease and drug drive optimal therapeutic development and management. This is the iconic matching the right therapy to the right patient. Characterizing pharmaceuticals through deep mechanism of action understanding enables comprehensive assessment of therapeutic effect and safety. We talked about that quite a bit. Personalized medicine implementation can enhance the pipeline and significantly improve patient care. And we talked about all these, basically the entire continuum. And lastly, strategic engagement with collaborators, whether academic or corporate, is key to demonstrate the value of personalized medicine towards successful delivery of solutions for patients. And before I answer any questions, I want to share with you that we are hiring, and you can look here at the features that we are interested in. As you can see, we cover a very wide range of activities along the continuum of personalized medicine, and we need people to lead programs both in the biomarker space as well as in the digital space. We're very innovative, and we're very much looking forward to maybe helping one of you come back home. Any questions?
Thank you, Iris. I think we'll give them a few minutes to write down their questions if they have any. Maybe if you can elaborate a little about the job you're looking to, the position you're looking to fill, specific projects that they might work on or anything. Okay.
So I'll talk about the two types of positions that we're seeking for. One is in the more personalized medicine biomarker space. We're looking for individuals with strong background. So as I said, PhDs or postdocs with strong background in different markers in genetics, genomics. We do a lot of whole genome sequencing or other types of genetics, genome-wide expression, proteomics, looking into metabolomics, looking into other newer modalities. Definitely imaging is a big focus these days. And looking at different types of tissues and media. You know, we have some markers that we're looking at in tiers. So it really depends on the utility. The individual is expected to be accountable for programs in mostly in the CNS space, where we have a compound that we're developing, and we want to answer all these questions that I mentioned earlier around mode of action, around responder populations, predictors of safety events, excuse me, and identifying new indications. The activities are very collaborative. We work with different groups, both in the R&D space, whether it's discovery, pharmacology, toxicology, and in the commercial space. In fact, there's a group, another unit in the commercial space called Diagnostics and Personalized Medicine, and they are our continuum. We work very closely with them to make sure that the biomarker or the signatures that we identify can be translated into a product in the market. And sometimes they come to us with a product, with a drug that's already in the market, and we're looking for biomarkers or algorithms that can help us differentiate it, make it more efficacious or less have less adverse events compared to competition. So that's the biomarker space. In the analytics space, we are looking to individuals that have a strong data science background, computer science, that can analyze or collaborate or guide analysts that look at big data, real big data, like sensor data, real-time, 18 months type of signatures that we want to correlate with different states, for instance, the movement disorder, and then use that as predictors on prescriptive algorithms that we will translate into medical devices. So being part of the whole development process, submitting to regulators, publishing, and also talking with payers. So it's a very, very diverse, very dynamic program with a lot of responsibility. I did get a question. Do you search for people in the field of electrophysiology, like in the case of NAV 1.7? So we are looking into electrophysiology markers. It is not as broadly used modality that I can use it in all my programs, but background in that is certainly useful. So it is certainly a possibility. I think I have another. regarding predominant and spine density, where are you going with this? Do you need to verify or validate this in human data or animal models? So obviously there are certain studies that we cannot do in humans that we do in animal models. In humans, you can look at ex vivo models and iPSCs, and including from treated patients, we look at different imaging modalities. But of course, some of the mechanistic data will remain in the animal model. There is no need to validate that because at the end of the day, the drug is approved by the efficacy data. And if it is efficacious, then it is interesting, of course, to know the mechanism, but the mechanism itself is not a prerequisite to the efficacy that you measure. Another question regarding the discovery of new treatments using pathways. Do you look for patient-specific pathways? If so, how computationally? So patient-specific pathways are relevant in the context of rare phenotypes, and we are looking into that. We have different collaborations to identify and recruit patients according to their unique phenotypes, and we're looking at different patient collections with different collaborators. But for the most part, when we look at pathways, the power is in the numbers. The concept of a individual pathway or individualized pathway is very difficult to develop a value proposition around in and of itself. So the idea is to be able to deduce enough from as many individuals as we can characterize, and the more we can, and the more the phenotype is well characterized and well ascertained, then we have more confidence in the data. Then that is useful to actually infer for larger populations, so that we can test and show the effect more broadly. Any more questions?
Okay, so I'll just fill in the space to let whoever is typing now not miss the opportunity to ask their question, and begin our thank yous and goodbyes. It was very, very interesting. I wrote it in the chat, but I will also say it. The webinar is being recorded, and we're loading it to our YouTube channel, the Science Abroad YouTube channel. And we had a question if we can share the slides themselves, Iris, so you need to answer that.
I believe so, but I'll double check and get that.
Okay, so once we'll have your answer or Deva's answer, then we'll send it or not send the answer to whoever asked it. So if anyone has any other questions, this is the time to press enter. And if not, I guess, again, thank you for being here, for sharing. Monica, thank you again. Yes. It was very, very interesting for me as a non-professional. It's my first view of personal medicine, so it was but I come from analytics, so it was very interesting. Thank you very much again for sharing. Thank you everyone for joining us and listening, and we'll see you next webinar, next month actually. Okay, so good night from Israel.
Thanks. Bye-bye. Night.
Bye.